So I'm Aviv Regev. I'm an associate professor in the Department of Biology at MIT, and I'm a core member at the Broad Institute, and I'm an early career scientist at the Howard Hughes Medical Institute. I was trained in both biology and computer science and math as part of an interdisciplinary program at Tel Aviv University, which is in Israel, where I was born and grew up. I also did my PhD in Israel, and afterwards I moved to Cambridge, Massachusetts, and joined the FAS Center for Systems Biology at Harvard as a Bauer Fellow. And there I had a small research group, and afterwards I moved to MIT as an assistant professor and then as an associate professor. I'm a computational biologist by training, which means that I try to combine methods and approaches that people develop in computer science and apply them to biological problems. And in particular, my motivation to go into biology actually came from computer science, because I think of biological systems, and especially of cells, as little computers that take information about the environment and try to process it and make all sorts of decisions. For example, should the cell live, or should it commit suicide, or should it make some type of molecule like insulin, or should it divide, or should it differentiate and become a different type of cell? So these and many other decisions are things that are very important for cells to get right. And for that, they take a lot of information about their environment, and they also have a lot of sensors about what goes on inside the cell. And then they try to process this information, decide what they should do, and execute this. And the way that the cellular computer works is by a lot of different molecules in the cell that interact with each other. Some of these molecules are proteins, some of these molecules are nucleic acids like our DNA and RNA, some of these molecules are small molecules like metabolites. These molecules interact with each other through physical and biochemical interactions, and this network of interacting molecules is essentially the cell's computer. Tony is right now trying to analyze transcription factor, which is a protein that regulates the expression of other genes, called SFP1. SFP1 regulates the levels of ribosomal protein genes, which are the genes that make up the different components of the ribosome, the machinery that translates transcripts into proteins. SFP1, which is an essential and important protein in the yeast, actually has very interesting evolutionary history. In yeast, it fulfills multiple different roles, but there are species in which this protein is present in multiple copies, and different copies fulfill different roles. This is one of the ways in which regulatory networks that control gene expression can change during evolution to assume different kinds of functions. So this is very convenient to study in the yeast because we can manipulate yeast genetically quite efficiently, more easily than we can in mammalian cells. And what we're trying to do right now is to test an antibody that, if successful, will let us measure all of the places in the genome in which the protein is bound in a single experiment. So as you might know, in biology labs, people pipette a lot and they move liquid around a lot. They use the pipettes actually to move liquid stuff from one place to the other. This can introduce a lot of errors and can also be very time and labor consuming. So what we see here is a liquid handler that's made by a company called Agilent. This is the Agilent Bravo. Down here fit things in a format that's very popular in biology called the 9612 format. And the liquid handler has some pins, which you might be able to see down there, that can go down into a plate if it's positioned here, pick up things and move them to other places and do it in a very precise way instead of a person actually standing and pipetting instead. And this is a nice programmable device. You can write code for it on a computer that's associated with it, so you can implement different types of protocols with it. So once you optimize the protocol and you know how you want it to run, you can actually let the machine run it to work. We can quite easily today, for example, sequence the DNA, and we can find in this DNA the different components that the DNA encodes in the cell. So, for example, the genes from which proteins are made. But it is quite difficult to understand how all of these proteins that are made, and DNA molecules and RNA molecules and many, many other entities all work together to actually process all of this information and compute a result. So to figure this out, we actually take an approach that comes from, that is motivated by how an engineer would think about this kind of problem. Sequencing actually underwent a huge revolution in the past decade, even less than a decade, in the past 6 or 7 years. And in this process, sequencing costs went way down, the throughput went way up, and the footprint of the machine has been shrinking. So now many labs, such as my own, but definitely many others at MIT as well, have these desktop sequencers, which, as you can see, are not a very big machine. They look like a slightly old and clunky desktop computer. And where a single machine like this in Iran might give you about half a gigabase of sequence. Now, the human genome is about 3 gigabase pairs, so you wouldn't need that many runs in order to get actually nice coverage of even a pretty large and cumbersome genome. And just for perspective, the Human Genome Project, the first time a human genome was fully sequenced, was an effort of about 10 years and billions of dollars. So that really illustrates how things have changed. Now, as a result, we now use sequencing in order to measure a lot of things about cells that before we wouldn't have used sequencing for. So we don't just use sequencing to sequence our DNA. We use sequencing in order to know where proteins bind DNA, how proteins interact with other proteins, which RNA molecules are in the cell, so which genes are expressed, where proteins are localized, which cells are living and dying. There are many, many different ways in which we can use these devices. So if you have a very complicated system that takes some inputs and needs to calculate some output, and you have no idea what goes on inside, it's like a black box to you. The first thing that you would do is that you would give it some information, some input. For example, you would put your cell in some condition, you will raise the temperature, or you will give it a growth factor. And then you try to measure something about what that cell did. For example, you want to look at which genes are turned on and which genes are turned off. Now, from this information that we measured, we try to take computational algorithms and reconstruct or reverse engineer what might have been the wires inside the black box that led the inputs into the outputs. Once we have that information, we have a model in our hand. But this model, we don't know if it's correct or not. It might actually be quite wrong. So what we need to do next is that we go after each entity in the model and we try to perturb it. So imagine if the thing that you were interested in was your cell phone and not a cell, and you were holding this cell phone and you had no idea how it works. The first thing that you would do is that you would punch the different buttons and you would see what happens. And now you might imagine that there are some wires inside and this is how they work. So now you will take the lid off and you will take something and you will manipulate the wires and you will try to punch the button again and see if it works in the way that you predicted. We try to do the same thing with cells. We actually go inside the cell using different types of molecular techniques and we try to silence the activity of different types of genes and measure again and see what happens. And if it fits with what our model predicted, then we tend to believe that our model was correct. And if it didn't fit, which is very often the case, then we go back again and we try to revise our model and we do again those perturbations, those manipulations, and we go again and again in a circle until we end up with a model that we think is well validated of how the cell works. Now, this is a question that's important for two reasons. The first is because we think it's actually really cool. We just want to understand how cells work. They're a fascinating facet of biology. But the second is that in our own body, there are many, many cells. And in most of our diseases, what happens is that these cells, for some reason, do not function properly. So in a cancer, the cell might divide too much or die too little. In an autoimmune disease, some of your immune cells might be overactive. And the way in which this happens is because something in this computer inside the cell is broken and doesn't work right. For example, it might be signaling all the time even though it shouldn't be. Or it might not be able to receive some signal that it should be receiving. So our hope is that by being able to decipher how the circuitry works, we can also identify which pieces of it are broken in a disease so that we can come into the cell and find a way of fixing the broken piece. Now, as you might understand by now, this kind of research requires skills and knowledge from many different aspects. So we need the experimental biology to help us actually measure and manipulate cells. but we also need computational tools in order to take the data that we collect and analyze it and try to build those models. And the two pieces really have to be connected very closely together. Working in MIT really helps us do this kind of interdisciplinary research, because in MIT we have both world-class biology but also world-class computer science and engineering. And these two disciplines at MIT are brought very closely together. So in my lab, for example, some of the people in my lab were primarily trained in computer science, but they end up working at the bench doing also biological experiments. And some of the people in my lab were originally trained in biology, but they also learn here a lot of computational methods and they apply them to the data that they themselves collected. And we also benefit from a lot of collaborations with other members of the MIT community, from the computer science department, from the physics department, from biological engineering, and of course from the biology department, from which we pick up a lot of methods and skills and also knowledge in specific biological systems.